Good. I guess afternoon, everybody. My name is Rachel Brasher, and I'll be giving my capstone presentation on establishing a system to evaluate the effects of ROS-induced DNA damage on mutagenic H-DNA. The most common form of DNA is the double-stranded B-DNA that can be seen over here. There's one strand in red and the other is in blue. DNA is also capable of forming alternative conformations such as H-DNA. H-DNA forms at mirror repeat sequences that can be seen here, with the 4 Gs on this side and 4 here. This sequence occurs about 1 in every 50,000 base pairs in the human genome. H-DNA is a triple-stranded conformation that forms when one strand breaks apart and twists back, binding to the 2 other strands, forming a triple-stranded region here and an available single-stranded region here. So you can see here, this is the single-stranded region. Previous studies have determined that H-DNA is mutagenic, leading to genetic instability in human diseases such as cancer and neurodegenerative diseases. H-DNA and alternative conformations such as H-DNA are recognized by repair enzymes as damage, and in an attempt to repair this damage, the repair enzymes will cleave the DNA, resulting in double-strand breaks. If this damage is not repaired and the DNA is replicated, it results in an altered sequence known as a mutation. One such mutation is a translocation mutation, where a part of a chromosome will break off and reattach to a different chromosome, as can be seen by this blue that was originally here and is now here. Same with the yellow. H-DNA specifically has been known to form near translocation breakpoints, where translocation breakpoints that overlap with genes in cancer. This is an example of how H-DNA leads to genetic instability in the development of cancer. Reactive oxygen species is another factor that is known to damage DNA. Reactive oxygen species are free radicals that have an unpaired electron, as can be seen by this singular dot. One specific free radical is hydroxyl radical. Hydroxyl radical is highly reactive and will commonly oxidize DNA. Specifically, guanine will be oxidized because guanine has the highest oxidation potential of all the nucleotide bases. When guanine is oxidized, it will form 8-oxo-7,8-dihydroguanine or 8-oxoguanine. Oxidation of guanine to 8-oxoguanine can result in transversion mutations, where instead of having a guanine cytosine base pair, we end up with a thymine adenine base pair. When this damage is not repaired, an accumulation of mutations such as transversion mutations will once again result in genetic instability. So now we can ask the question, how does oxidative stress affect H-DNA-induced mutation frequency? Our hypothesis is that the H-DNA structure, specifically the single-stranded region, is more susceptible to DNA damage to control B-DNA sequence. This hypothesis is based off the knowledge that the single-stranded region contains a significant amount of guanine bases, which, when under oxidative stress, the guanine is readily oxidized. The overall objective of this study is to compare the H-DNA and B-DNA mutation frequencies in the absence and presence of oxidative stress. A yeast artificial chromosome containing either an H-DNA forming sequence or a B-DNA forming sequence will be used to compare the mutation frequencies. In order to induce oxidative stress, ROS must be added. H2O2 or hydrogen peroxide is a molecule known to form the hydroxyl radical, so that's what we've decided to use. This overall objective was determined based on the knowledge that B-DNA um, is, is more mutagenic under oxidative conditions than standard conditions, which is without hydrogen peroxide. So the protocol to compare mutation frequencies is a HDNA forming sequence YAC and a BDNA forming sequence YAC is added to solid media and grown in that, and then stressed with hydrogen peroxide and liquid media. So we have HDNA or BDNA either in the absence of hydrogen peroxide, so without, or in the presence of hydrogen peroxide with it. Um, Colonies are then grown on two different plates, one that will only allow for the mutants to grow, and one that allows for total colonies to grow. Mutation frequency is then calculated as the mutants over the total colonies. But before this experiment can be performed, a hydrogen peroxide concentration that will not result in a significant amount of cell death first needs to be determined. So in order to determine a concentration of hydrogen peroxide that does not result in a significant of cell death, I adapted a protocol from Tran et al. Once again, a YAC containing either an HDNA or a BDNA forming sequence was grown in solid media and then exposed to hydrogen peroxide in liquid media. I decided to use 0 millimolar hydrogen peroxide as the control non-stressed condition, and then use 0.2 to 20 and 200 millimolar hydrogen peroxide as the stressed conditions. After being stressed, cells were serially diluted in water, plated, and then allowed to grow for approximately 2 days. Percent survival was determined as the stressed, so the 0.2 to 20 and 200 millimolar over the 0 non-stressed millimolar, and then multiplied by 100. So I was able to determine that approximately 2 millimolars of hydrogen peroxide does not result in a significant amount of cell death. This was determined by finding the lethal concentration. So the lethal concentration is the concentration of hydrogen peroxide that correlates to about 50% survival. 50% was chosen because it is a good balance between too much stress, or that will result in cell death, complete cell death, as is seen by 0% survival, and too little stress, where there will be no effect on the cells, which is evident by 100% survival. As you can see, 20 millimolar and 200 millimolar resulted in complete cell death, and 0 millimolar resulted in no effect at all, which we expected since there was no stress added, so we do not expect cell death to occur due to stress. Now, as we can see for HDNA, the blue line, the 50% survival was found to be approximately 2 millimolar. Similarly, with B-DNA in the red line, we found that it also correlates to about 2 millimolar. So now we can ask the question, does ROS increase HDNA mutation frequency? We can perform the previously stated protocol and add the approximately 2 millimolar hydrogen peroxide. We expect our results to be, so previous studies have shown that under standard conditions, HDNA has a higher mutation frequency than the control BDNA. HDNA is in the red and B would be in the blue. HDNA was seen to have a 9.4-fold higher increase or higher mutation frequency than the BDNA. So under oxidative conditions, we would expect that the B-DNA and H-DNA mutation frequencies would increase, and we would expect that this fold would also increase. Um, and I would like to thank Dr. Zoel Foote for mentoring me in the lab, Ashley Shavana for working in the lab with me, and Mariah and Elijah for moral support over these last couple of weeks. I'd also like to thank the NIH, the Welch Foundation, and of course, Southwestern University. And that's it. Thank you.
Thank you, Rachel. Do we have any questions? Let's see who's raising hand.
Where's the raise the hand function again?
I think it's under participants.
Participants. Let's see.
I'm not seeing it there. Oh, Professor Delino found it. Okay.
Delino has Drew.
It's on the bottom right of the participants box, Ethan. But you can ask your question first. Go ahead.
Oh, thank you so much. So Rachel, you did fantastic.
Thanks, man.
You're welcome. So I was curious, this is like probably tangentially related and probably isn't correlated, but I was curious, like with methylation, can methylation affect, I guess, like H-DNA formation?
You have like a more specific, like, I don't know, I guess you don't know the structure of H-DNA. So yeah, I don't really know. I guess it probably would.
Okay, cool. Yeah, I was just curious. My biochemistry knowledge is not as good as it once was. Thank you so much.
Mm-hmm.
Okay, Dr. Delino, go ahead.
Yeah, hi, Rachel. Great job. I thought it was very interesting. I had a question. So if I remember correctly, you said that the single-stranded region, the non-triplex strand, was what more susceptible because it had more guanines. But then in your image, the mirror repeat and the triplex region in your figure describing H-DNA had more guanines in that triplex region, and the single-stranded region didn't. So, figures are just sometimes representative and just showing a typical thing, although not necessarily. But is it typical for the single-stranded region, the strand that's not part of the triplex, to have more guanines, or just in your particular system that you were testing?
So I think it's common for the single-stranded region to have a significant amount of guanines. I don't know for sure if it has more guanines than the triplex region, but the mirror repeat sequence also tends to have a significant amount of guanine bases, and so I think that's why it's shown in that figure. The guanine bases there. So as a whole, when people look at H-DNA with mutagenesis, there's hypotheses that believe that the triple-stranded region is more mutagenic because of the guanine basis, but our hypothesis is that it's the single-stranded region. And so they both have a significant amount of guanine bases. If that.
Okay, great. So it's more CG rich rather than guanines partitioning or being more, okay, in the single versus triplex. Okay, great, thanks.
Mm-hmm. Let's see, Dr. Wiegand.
I think you might be muted.
He's muted. Dr. Wiegand, you're muted.
Try it again. Okay, here we go. How do you identify whether the cells are mutated or not? Is there some physical or visible mode that you look at
to determine where they're mutated, like within the sequence?
No. If they have mutated, or do you just do it by chemical sequencing?
So, if we go back here, we'll go here. So for this, this plate right here will only allow mutants to grow. And it does this by, so FOA the addition of FOA. When the cells are mutated, they do not have an active URA. Let me go back to that. So here, in the yak, when the cells are mutated, this URA is inactive. And so when grown on FOA plates, mutants that do not have the URA will grow. But if they're non-mutated, then the URA will react with the FOA and form a toxic molecule that's called fluorodeoxyuridine, and it will kill the cells. So the non-mutants are unable to grow on that plate. And so that's how we get the mutants.
Oh, okay. Thank you.
Let's see, any other questions for Rachel? We have a couple more minutes.
Sorry, if we do have a couple of minutes, I had another question.
Yeah.
Rachel, great job again. Translocation mutations, do they require H-DNA being in both positions? So for example, like the Philadelphia chromosome, the mutation that causes BCR-ABL is a translocation mutation between, I don't know, I think chromosome 8 and 22, I'm not sure. But does that require that you would have H-DNA in both positions, or would an H-DNA in one position simply be more susceptible to translocation randomly to any other position in the genome?
I believe H-DNA only has to be, when you say the two ligations, you mean the two chromosomes?
Correct.
Okay. I believe that H-DNA is only has to be on one chromosome, the sequence for it. So it's, I believe that, so H-DNA would be like here, and then a sequence that specific, if we're talking about the translocation mutations that have to do with cancer, then one would have the cancer gene and one would have the H-DNA forming sequence, and then they would switch. So I think H-DNA only has to be on one chromosome.
Okay, great, thanks.
Hey. Thank you very much, Rachel. So I think now you have to make Mariah host. Can you do that?
Yeah, I think I
On the
click
three
here.
dots in the rectangle.
Here?
Dr.
No.
Whalefoot has a question before.
Okay. Oh, sorry about that.
No, I was just applauding.
Ah.
Thank you.
I'm trying to give you a hard time, Rachel.
Dr. G, I'm kind of surprised you didn't ask me any questions.
So you can, maybe you can escape, I don't know, from your presentation, right? And now go to the picture of Mariah, and then in the three dots.
Where's Mariah? There's Mariah.
Just there.
Ah.
You can make her perfect. Awesome. And Mariah, you can start whenever you want.
Okay, can everyone see my screen and hear me? Okay, I'm assuming that's yes.
Yes.
So good.
Yes.
Okay, sorry.